I am very happy to announce our next speaker. So this is Dr. David Simon. He's a professor of neurology at the Harvard Medical School and chairs the International Linked Clinical Trials Committee, which is something by us and the Andel Institute. And we'll talk a bit more about that later today. So his research focuses on all things Parkinson's, especially mitochondrial dysfunction, which is what he'll be talking to us about today, specifically mitophagy. So I will hand it over and share your slides.
All right, thank you very much. Yeah, I'm very happy to have a chance to talk to you today. It's not often I talk about a topic like mitophagy to, a group that's not all scientists, but I hope to tell you why this is relevant and maybe of interest if you're hoping to ultimately slow progression of Parkinson's. These are my disclosures. Actually, there's one thing I think I added to it, but not on this version of the slide, is that I also did some of the work I'm going to talk about was in collaboration with Mission Therapeutics, which is pushing forward a therapy that I'm going to talk about. So, questions for today. What is mitophagy? How is mitophagy related to Parkinson's? And then I'm going to talk about USP30, which is a target that we're interested in the lab, and I'll explain why I think that might be of interest if you want to ultimately slow progression of Parkinson's. I'll talk about how this molecule blocks mitophagy, and then, well, at least give the beginnings of an answer to the question of whether or not blocking USP30 might slow progression. So this slide is just to point out my interest in, neuroscience and neurology is because of this amazing structure of the brain, which results in things like cognition and consciousness and all sorts of amazing tasks. So how did I go from there to an interest in this little subcellular organelle, the mitochondria? But it's actually relevant, and that's what I'm going to talk about today. So, beginning question: what is mitophagy? The word comes from the word mito, or mitochondria, which are the energy factories of our cells. Chemical energy is ATP, which is like a chemical stored form of energy that's used to do everything cells need to do, and a lot of the cellular energy is made in the mitochondria. And then phagy, from the Greek word eat. I think this is a Joey Chestnut from the Nathan's hot dog eating contest. Not a contest I recommend. So that's okay. So in any case, so mitophagy is the degradation of dysfunctional mitochondria. So I'll talk about the concept of mitochondrial turnover today. We all have these energy factories in our cells, but over time they become dysfunctional. Oh, and the time I'm talking about here is on the order of days, so they have a relatively short half-life. And when they become dysfunctional, you have to get rid of them. Meanwhile, you're making new, fresh mitochondria, and that's how you maintain a healthy pool of these energy factories. So what happens when mitochondria don't work right? Well, one is you get less of that cellular energy produced, the less ATP. Another is a byproduct when mitochondria don't work right is that it uses something called the electron transport chain. And when these electrons don't go to form ATP, they kind of go in the wrong direction and form these highly reactive molecules that basically react with and damage any nearby molecule. Could be DNA, lipids, or protein, and that's free radical damage to these other proteins, oxidative stress. So dysfunction causes loss of energy, ATP, and oxidative stress. So there's a lot of evidence that mitochondrial dysfunction is there in Parkinson's, and I'm not really going to talk so much about that. But the question is, how do we know that that's important? I mean, you can have loss of energy just as a byproduct, as cells are dying of some other cause, or it could just be incidental. So part of the answer that why the mitochondrial dysfunction actually might be relevant to the cause of Parkinson's, one of the first things came from this story, The Case of the Frozen Addicts by Bill Langston and a journalist, John Halferman. They noted that in California a few decades ago, showing up in the ER all of a sudden were many young drug addicts who looked like a person with advanced Parkinson's, except they'd been fine a few days earlier. It all came on for just a few days, very rapid onset. And it turns out he did detective work, Bill Langston, to help track down what happened. And they synthesized a narcotic derivative that turned out to be MPTP. And this MPTP has a metabolite that is taken up specifically by dopamine-producing neurons and blocks mitochondrial complex 1, one of the subunits involved in that energy production. So now they've shown that specifically blocking mitochondrial function kills dopaminergic neurons and causes something that looks a lot like Parkinson's. One problem here, though, is, I shouldn't say problem, but one, it doesn't explain necessarily that mitochondrial dysfunction selectively kills dopaminergic neurons, because in this case, the toxin is taken up only by the dopamine-producing neurons. But Tim Greenemeyer, who we're going to have the honor of speaking with shortly, did a really important experiment that's been very highly cited and influenced my own work, among others, and that's this study where he looked at a common pesticide called rotenone that is actually used in laboratories as sort of a control because it's very specific at binding to and blocking mitochondrial complex 1. So at high doses, it's lethal, but at low doses, exposed chronically, in this case to rodents, he was able to show that they would develop a gradually progressive loss of dopaminergic neurons. So here's another mitochondrial toxin that causes changes that look like Parkinson's, but in this case, the difference is it's not taken up by the dopamine transporter. This causes this mitochondrial dysfunction in all cells, not just in the brain, but throughout the body, yet preferentially the dopamine-producing neurons die. So there's a sign that these dopamine-producing neurons are preferentially vulnerable to this mitochondrial dysfunction. I'll talk about why in a moment, but interestingly, followed up on this was an epidemiologic study led by Carly Tanner, also Bill Langston was involved here, that showed that if you can measure exposure over life to rotenone or related pesticides or herbicides in the environment, paraquats and herbicide that also affects mitochondrial function. And people with higher levels of exposure to these pesticides have a higher risk of developing Parkinson's. So interestingly, rotenone was considered an organic compound. Organic because it's natural, it's plant derived. So yeah, I mean, this just to make the point that natural isn't always good, right? So it's a natural compound, but it selectively kills dopamine-producing neurons if you get it in the right dose and quantity over time. So thanks to Tim Greenemeyer's work, this was no longer considered okay for organic farming. However, it's still used. And here's an example of one study, this is from a while ago, though it's still used today, where they were looking at this small lake that had some invasive fish in it. So they had a simple plan, just dump 17,000 gallons of rotenone. I mean, I'm imagining how careful we are using this in the lab, with gloves and masks and measuring out minute quantities to use in our experiments. And these guys are dumping thousands of gallons of rotenone into the lake. And of course, all the fish die and float to the surface. And sure, it's a source of drinking water for this downstream town, but they assured everyone there was no reason for concern. So alpha-synuclein is one of the pathological hallmarks of Parkinson's. This is a protein, we all have it, but it actually builds up in the brain in people with Parkinson's. And when it forms these aggregates that you can see in a cell under a microscope, we call that a Lewy body. So alpha-synuclein is a major component of Lewy bodies, and too much alpha-synuclein contributes to Parkinson's. There's a lot of evidence for that, genetic and otherwise. And it's, the genetics confirm that it's a cause of Parkinson's, and then it looks like it's relevant even to sporadic Parkinson's, where they also develop Lewy bodies, at least most patients do. I'm talking about mitochondria today, so the connection of alpha-synuclein to mitochondria, interestingly also from Tim Greenemeyer's work, and this is not just because I'm about to be interviewing him, these slides would be in my talk anyway. He did some important work showing that alpha-synuclein, there was other work generally showing alpha-synuclein causes mitochondrial dysfunction, but he showed specifically it binds to this protein importer on the surface of mitochondria called TOM20. And this is my very crude attempt to illustrate what I'm talking about here. So sorry, pointer's not consistent working. There we go. So TOM20 is like a doorway on the surface of mitochondria. Proteins that need to get into the mitochondria to function have to go through this doorway, TOM20. Alpha-synuclein is like a bouncer at the doorway, stopping the proteins from getting in. Now, those proteins are synthesized in the cell outside the mitochondria and have to get into the mitochondria in order for the mitochondria to work right. So alpha-synuclein is starving the mitochondria of these necessary proteins and causing dysfunction. So what happens then, when you have these mitochondria that aren't working, if mitophagy fails, and I'm going to talk about why we think why mitophagy fails in Parkinson's, at least one example of it. When mitophagy fails, if you think of the mitochondria as the trash you need to take out, it builds up. This is, I think, a Philadelphia garbage strike. So again, think of it as a turnover. You have healthy mitochondria that need to consist, all the time being synthesized. So you have a new healthy pool of mitochondria. But part of that the important part of it is you also develop mitochondrial dysfunction. You have to degrade the dysfunctional mitochondria. So that's an important homeostasis. They both need to happen. I'm not going to talk about it today, but our lab also is interested in the mitochondrial biogenesis side. There's a molecule that regulates mitochondrial biogenesis called PGC1 alpha that seems low in Parkinson's, and that low PGC1 alpha may also be important. But today we're talking about the other end, the mitophagy. So before I get to that, just a little aside, why dopaminergic neurons, why are they preferentially vulnerable to energy dysfunction? I mean, pretty much all of our cells have mitochondria, not quite all, red blood cells don't, but almost all our cells have mitochondria, and they depend on that to survive. So why dopaminergic neurons? Well, one is, here's a drawing of two axonal arborizations. So when neurons make connections to other cells, they send out the processes that branch out and make the connections that neurons need to talk to each other. For dopaminergic neurons, those are huge. They make massive arborizations, and also they have pacemaking activity. So they're constantly firing at which is termed pacemaking. And both this constant activity together with these massive arborizations leads to huge energy demands. So one idea is they're the first to go if you develop an energy deficit because of mitochondrial dysfunction, because they really depend on this. So how do you know when a mitochondrion is not working right and you need to throw it away? I mean, here's an example of a bag. Maybe that's trash, maybe it's not. How do you know if this is something you want to throw away? Well, you label it. Stick a label on it that says trash, right? Now you know, okay, that's a bag of trash, you want to throw it away. Well, in our cells, we have a couple of proteins, one called PINK1 and one called Parkin, that cooperate specifically for the purpose of identifying dysfunctional mitochondria and putting a trash label on it. The trash label is ubiquitin. That's a cellular tag that's put on the mitochondria, and when you have ubiquitin on mitochondria, that's the signal that it needs to be transported to lysosomes, which are the trash bins of our cells where they get degraded. So PINK1 and Parkin, you can see from up here, are highly penetrant causes of Parkinson's. They're rare mutations in those that cause early onset Parkinson's. So, I mean, often onset in the teens or 20s. And they're loss of function mutations, and they're autosomal recessive, meaning you have to inherit one mutation from one parent and a mutation from the other parent. So you have almost complete loss of function, or maybe complete loss of function of those genes in order for it to cause Parkinson's. So they both play key roles in mitophagy by labeling the dysfunctional mitochondria. And loss of function, therefore, of either of these genes leads to impaired mitophagy. Other genetic factors I'm not talking about today that are where mutations are associated with Parkinson's also seem to play a role in mitophagy. So conclusion is that this impaired ability to degrade dysfunctional mitochondria, again, is not just a downstream consequence, it's actually a cause of Parkinson's. So that leads to the question, well, maybe we can slow or stop progression by enhancing mitophagy. So how can we do that? One way is we can look at a molecule called USP30. This is what I mentioned earlier. USP30 it's called a deubiquitinating enzyme, meaning it removes ubiquitins from other proteins. Remember, ubiquitins, I said, are that trash label. So Parkin and PINK1 are putting the labels to identify mitochondria that need to be degraded. USP30 is removing those labels. So inhibit USP30. So that's what we test it. We wanted to validate whether or not USP30 would be a good target for Parkinson's. So we did it in two ways: a genetic manipulation, which is where we actually knocked the gene out of the mouse. So you actually make the mouse model where it has no USP30. Very specific. You know it's only that gene being targeted, but also not very translational. We can't knock out the gene in humans. But also, with collaboration with Mission Therapeutics, did a small molecule to test and see if a small molecule that inhibits it could work. And that's more interesting from a translational perspective, because in theory, you might be able to take that into the clinic. So let me tell you a little about the results. So first, what happens if you knock it out? I mean, if that killed the mouse, that wouldn't be good, because that would tell you, okay, you have to be careful with this molecule. But it turns out the mice look healthy and they live a completely normal lifespan. It's just showing how long mice live who don't have USP30 compared to those who do, and it's identical. So that's encouraging. A complete knockout doesn't cause any gross or overt harm. That's encouraging that a partial inhibition with the drug might be safe to do. And then remember, we predict that knocking out USP30 will increase mitophagy, because you prevent this counteraction of what Pink1 and Parkin do. And we did show that here. And this is the mice also had a special fluorescent signal, which I can talk about later if you're interested, which allows us to track mitophagy, and it's increased. And this is specifically looking in dopamine producing neurons. So knocking out USP30 did achieve the goal of increasing mitophagy. And what does it do in terms of relevance to Parkinson's? Well, we don't have a perfect model for predicting what's going to work or not work in humans, or maybe we do, but we don't know it yet, because we need to get more success in patients to know whether or not the mouse models predict it. But I think one of the better ones is where you make a mouse express too much alpha-synuclein in the dopamine producing neurons of the substantia nigra. And if you do that, that kind of mimics what we know happens in humans. Some humans have mutations that cause them to produce too much alpha-synuclein, and they get Parkinson's. So it's probably a good model that has relevant mechanisms. And we do that using basically gene therapy in a mouse, where you inject a virus on one side of the brain, and they develop slowly over several months loss of, no, the pointer's not working, but here you can see loss of that dark pigment area is the dopamine producing neuron terminals in its target region called the striatum. And you see a lot less staining on the left-hand side, the side where it expresses too much alpha-synuclein compared to the other side. But on the right-hand side, you see in the USP30 knockout mice, it looks more similar on the two sides. So USP30 knockout prevents the alpha-synuclein-induced loss of those dopamine terminals. And then if you just count the neurons, you use something called TH, which stands for tyrosine hydroxylase. We have antibodies that specifically bind to it, and we can use those to see the neurons that make dopamine, and we can count them under a microscope. And if we count those neurons, you see compared to normal, you have more than a 50% loss of the neurons caused by the alpha-synuclein. And again, we can use the other side of the mouse as the control. But in the USP30 knockout, you have a lot less loss of those neurons. So it protects you not having USP30, so that you have increased mitophagy, protects against loss of neurons induced by alpha-synuclein. And what about the alpha-synuclein itself? Well, here, there we go. Okay, so here you see you have this pathological form of alpha-synuclein, a phosphorylated form at serine 129. You see very little of this form of alpha-synuclein in the control mice. In the mice that have overexpression of the alpha-synuclein, you see much higher levels, almost complete elimination, pretty dramatic, robust decrease in the cells that have the enhanced mitophagy. Our idea on this is, remember I said the alpha-synuclein is binding to the mitochondria at the TOM20 and causing mitochondrial dysfunction. That then triggers, when you lose USP30, you're now triggering the degradation of those mitochondria. Well, the alpha-synuclein is going to get degraded along with the mitochondria. And maybe that's why we have such a powerful decrease when we enhance mitophagy. So loss of USP30 leads to normal lifespan. That's good. Increased mitophagy, clearance of alpha-synuclein, and protection against alpha-synuclein-induced. I guess I didn't don't think I showed you the behavioral deficits, but you can look at use of the forelimbs and show that it's the asymmetry that's caused by unilateral alpha-synuclein injections is prevented by this, completely prevented. So I think this validates that USP30 is a good target. We want to inhibit USP30 as a potential neuroprotective strategy in Parkinson's. But again, we did this with a genetic manipulation. So with this in mind, Mission Therapeutics is one company. There's one other I know of also developing these. Is developing a small molecule inhibitor, and they have a lead candidate that can be given orally. That's important. Not all drugs can, but this one is orally absorbed. And it does get across the blood-brain barrier. Again, not all drugs do. Some drugs get into the blood, but don't get into the brain. This one does. And they validated it in cells and mouse models that it does increase mitophagy, similarly to what we showed you with the knockouts, and protects against the loss of dopamine and the loss of dopamine-producing neurons. So it replicates a lot of what we saw with the genetic knockout. And Mission Therapeutics now has this MTX325 in clinical trials. It's in phase 1 clinical trials, although it has been for a while now, and I guess we're waiting to hear an update on what they've learned there. But and that's great, because a lot of drugs that look, or a lot of targets that look promising, it's hard to get a drug into clinical trials. There's a lot of steps you need to do to get FDA approval to do a trial. So it's a huge step to actually, even though it's an early phase 1, huge step to get there. On the other hand, phase 1 is still a long way from where we want to be. Assuming they're successful and it looks safe and tolerable, which I'm optimistic on based on the prior studies, then you got to do a phase 2 with larger numbers of patients to get more data on safety and efficacy, and then ultimately a definitive larger phase 3, and those would be placebo-controlled, whereas the phase 1 generally are not. And then hopefully FDA approval. Now a lot of drugs fail along that process, but this is one I think we have reason to be optimistic about. So conclusions: mitochondrial dysfunction contributes to Parkinson's. Impaired ability to remove dysfunctional mitochondria also seems to contribute to Parkinson's. USP30 inhibits the mitophagy, but blocking it, either by genetic manipulation or with the drug, improves mitophagy and protects against alpha-synuclein toxicity. So based on this, we think inhibition of USP30 is a promising strategy with clinical trials underway. And sorry, I skipped over the last one. But one of the last thing I want to mention is that this MTX325 was reviewed by the International Link Clinical Trials Committee, Cure Parkinson's, and was prioritized as a high priority agent to move forward based in part on some of the reasons I've shown today. So we're all very excited it's kind of made it into clinical trials. And I know there's another, Vincere Biosciences in Cambridge is also looking at USP30 inhibitors. So there's multiple paths we can keep a watch on. And happy to have had a chance to talk to you about this today, and would welcome any questions. Yeah.
Is this just a subset of people with Parkinson's have this particular dysfunction, or is this dysfunction, mitophagy, common to everyone with Parkinson's?
Yeah, really important question, and sort of a complex answer. So I talked about some people where clearly they have dysfunction, not just of mitochondria, but of mitophagy, the rare people with PINK1 or Parkin mutations. But that's a very tiny percentage of people with Parkinson's. However, there's much broader evidence for mitochondrial dysfunction in most people with Parkinson's, and alpha-synuclein pathology, which is present in most people with Parkinson's. We think anybody with the alpha-synuclein would probably benefit from USP30 inhibition because it can help counteract the mitochondrial dysfunction that alpha-synuclein causes and helps clear away alpha-synuclein. There are lots of strategies aimed at clearing alpha-synuclein, and although this one does it indirectly through targeting mitophagy, I think it would be a benefit to most. The complexity comes when there are some people who look at various ways to find biomarkers of mitochondrial dysfunction in patients, and they find only a subset of patients have those biomarkers. But at this point, I don't know that we should just target those people. I would imagine in an early clinical trial, my view, at least based on what we know now, might be include a broader group of people with Parkinson's who have evidence of alpha-synuclein pathology. but measure these biomarkers. And maybe in the phase 2 trial, you might learn that, yeah, it does look like a subset are more likely to respond, but I predict maybe it'll be more broad in terms of the responsiveness.
You mentioned that PINK1 and
Parkin, yeah.
Parkin were associated with early onset. That kind of interests me for some reason. Are you saying that everyone, more or less everyone with early onset has that PINK1 and Parkin gene problem?
Yeah.
Can I butt in here? This for me is one of my bugbears, because there is a trend for everyone to call early onset Parkinson's, and that gets in a muddle with early stage Parkinson's versus young onset Parkinson's, where people are under the age of 40. And the whole field keeps on saying early stage Parkinson's, and it's just like, oh my goodness, that's going to be confusing to the community. And Dan, you've just proved my point. Thank you very much.
Okay, so I apologize. I think I call it hopefully early onset, not early stage, but still young onset's better to avoid confusion. Yeah.
It's about age rather than, yeah.
Yes, early is an ambiguous term. So yeah, I like young
really
onset,
is,
so I'll try to remember that. Yeah.
Particularly given that the whole research field in general is trying to go in earlier in the stage of the disease and trying to be treating people before they become, have complication motor symptoms. Young is important. We're young at heart.
So we call it what?
Young onset.
Young onset.
Yeah. So the answer to your question though is no, not every young onset patient has one of these mutations. Or I think for the very young onset, like before age 20, there's I think it's more. You see different numbers in different studies, but maybe more than half would have Parkin gene mutations.
But what about Michael Fox, for example?
So he's young onset, but not so young. He was, I think, around 30 when he started. So the percentage who have a Parkin gene mutation is much higher in somebody who gets onset as a teenager. In the 20s, it's still high, but not quite as high. In the 30s, you're down to, I think, just a few percent of those. And so there are others that I didn't talk about that are rare. DJ1 is one, BPS35, and others that are rare causes. All those. And then there's some people who have young onset cases, and we do the screen and we don't find a mutation. So it's not all, but it's and it's still pretty uncommon. I follow one patient who has Parkin gene mutations, and I diagnosed her at age 19. She'd already had symptoms for 3 years by then, and she's now 32. I'm still following her, and doing well at times, but some issues. So, yeah.
I'm curious how you begin to identify candidates for research on this, especially as you begin doing phase 1, phase 2, phase 3, because it sounds like it's not a simple external test. You actually have to go in and do some kind of genetic marker in that. So how do you begin to identify your candidates for research?
Yeah, so I mean, for phase 1, it may be less critical, or in terms of the inclusion criteria. But when you get to phase 2 and phase 3, that becomes a really critical question related to what was asked before. With precision medicine, you want to identify people who have the specific problem that you're targeting and only treat them. On the other hand, if you have a treatment that would help almost everybody with Parkinson's, that's even better, although some people think you're going to have a greater chance of success if you can do the precision medicine route. For this one, my answer is I don't, beyond showing evidence of alpha-synuclein pathology, I don't think we have enough knowledge to use these markers to specifically target people. So if you did, in this case, I mean, early stage Parkinson's patients, they're commonly used for trials trying to slow progression, just because it's easier to follow the clinical markers of progression that we try to follow. You'd probably include anybody who meets the general eligibility criteria, not select those who have a marker. But it may turn out that those who have those protein markers of mitochondrial dysfunction in the periphery, which we can measure, or an imaging measure of mitochondrial dysfunction in the brain, might turn out to be the ones who respond best. And if we saw evidence of that in the early trials, in the later trials, we do exactly what you said. There is a kind of imaging where you can look at sort of high energy versus low energy phosphates in the brain as one sort of indirect measure of mitochondrial function. There's also, protein-based biomarkers in the blood you can look at, and maybe we'd end up choosing people based on, these initial blood tests or imaging tests.
What, so would these persons involved then be levodopa? Would that would skew your result, right?
So, yeah, I think the question you're asking now are really important questions that apply to any clinical trial where you're aiming to slow progression, not specific to USP30 as a target. And every time we design a trial, we have numerous questions, any one of which sort of could be the key to success or failure, but are really important to consider. And you're raising one of those. What do you want to enroll patients before they're on levodopa? And of course, that has the advantage when you rely on clinical markers, because levodopa can mask some of the symptoms that you're trying to measure. But then a lot of patients early on, even at diagnosis, might choose to start levodopa, so you're excluding a lot of patients from your trial if you do that. So there's a lot of mixed factors, and you could also think about, well, what would be the ideal population, but it might be such a small subset that it's not feasible to find enough patients. Often clinical trials are struggling to get enough patients who are eligible and able to participate. So you kind of always have to make some compromises over scientifically ideal versus feasible. But these are questions that'll be debated later, I hope, for USP30 inhibitors. Yeah, sorry.
So from my understanding, LRRK2 variants also have mitochondrial dysfunction, but about half of LRRK2 variants have no alpha-synuclein pathology. So is that a different type of mitochondrial dysfunction then versus this USP30? And then how would you distinguish between what type of mitochondrial dysfunction, like on your biomarkers?
Yeah, you mean in LRRK2 patients? Yeah. So LRRK2, I think if you're thinking maybe about the alpha-synuclein seeding assay, which is where you can measure a pathological form of alpha-synuclein in the spinal fluid. Michael J. Fox Foundation helped to develop this. I think it's about a third of people who have a LRRK2 mutations test negative on that. Actually, I learned from Dr. Greenemeyer, who's joined us here in the back just now, that there may, we don't have fully understand how that seeding assay correlates with brain pathology, and it may not correlate perfectly. And my understanding, though I don't know the details, is that there is some evidence that even those patients who test negative on the seeding assay may have a form of alpha-synuclein pathology, and vice versa. So,
like those recent papers on the oligomers,
yeah,
looking at maybe it is a synucleinopathy. It just, we can't see it yet.
Yeah, that's an important question, though. When I talk about wanting to enroll patients for a USP30 trial who have evidence of a synucleinopathy, we could use the seeding assay and say only enroll people who are positive on that, but we might be excluding patients who are equally have equal potential to benefit because they might have pathology in the brain, but not in the seeding assay. So we, by the time we get to that trial, we may have more information about this and can adjust accordingly.
And perhaps it's a matter of taking everybody, but just doing all of these measures in that cohort so that you know what people have got, and then learn from that for the future studies.
That would be I agree with that strategy. That's what I think would be best, unless our knowledge progresses, so we can do better than that from the start. Okay.
Thank you, David. That was